recording. And here we go. Okay. So lesson 10 on protein synthesis. So we're going to continue. Oh, great. And now here is Nagar. Okay, fantastic. Everybody is here. Sorry about that. All right, so today's lesson on protein synthesis, we talked about, in our last lesson, we talked about some of the experiments that the various scientists had done that brought us now to our current understanding of the structure of the DNA molecule. But really what happens is there's more to just DNA and DNA replication and how genetic material is copied and passed on in cells. It actually, what DNA really is the blueprint, the instructions to the working of the organism as a whole. So in our last lesson, we focused on that genetic information and how it's stored, how it's copied, how it's passed down, how mistakes might get corrected. In today's lesson, we're going to talk about how proteins are made using the instructions that are encoded in DNA. So we are going to talk about all the relevant terminology, and hopefully by the end of this lesson, you will learn how to apply these all correctly. We will talk about terms such as mRNA, tRNA, rRNA, the term codon, anticodon, transcription, translation, and we'll also talk about the ribosomes. We'll also look closely at a strand of DNA, for example, to look at the genetic code and how the base pairing is determined by the sequence of the DNA and how that sequence gets translated to protein, and also figure out what happens when there is anomalies and mutations along the way. And we'll also look at the different various steps that are involved in going from DNA to protein, and how that gene expression is controlled both in prokaryotes and in eukaryotes. And so as we're doing this, we're going through our lesson, you can think about some of these questions, such as what is the role of RNA in protein synthesis? How is genetic information transformed finally into an actual working protein and to an enzyme? And how do mutations in the DNA code affect the final protein product? So really where we are right now in our understanding is we know today that basically one gene is the code, the instructions to create one polypeptide, but this wasn't always the case. And there was various discoveries and various experiments done along the way to arrive at this hypothesis of one gene, one polypeptide. And the first part of this process was the work that was laid out by Gregor Mendel, which we talked about a little bit in last class. And essentially what his work, just to recap, was that it established that there were some factors that played some sort of a role in the transmission of traits from parents to offspring. And he showed this in his pea plant experiments. And we know now today that these factors are actually genes, and that they, what the genes do is that they provide instructions for the production of proteins who have specific functions in the cell, and those specific functions eventually get expressed as phenotypes or the physical traits that we see that are passed down from generation to generation, from parent to offspring. And so this idea was a little bit further continued by another person called Archibald Garrod. And Archibald Garrod was a doctor, and he noticed through his work that there were some illnesses that seemed to be present in many members of the family and were kind of passed on in the family. And so he hypothesized that there must be an error in one of the enzymes that could create this illness. And he called this and sort of an inborn error of metabolism. And he proposed that there were the enzymes that had to be under the control of these hereditary material. And so therefore, if there was an error in the hereditary material, that error led to an error in the enzyme. So for example, he hypothesized that if there was, for example, a metabolic pathway where you had reactant A processed through enzyme 1 becoming B, processed by enzyme 2 to become C, processed by enzyme 3 to become D, the final product. For example, if you have an accumulation of substance B, this must mean that enzyme 2 would be defective because enzyme 2 takes care of transforming the substance B to the next substance in the pathway. And so if you have a buildup of B, that means that this enzyme 2 must be defective. So he hypothesized that these hereditary material direct the production of these enzymes. So he linked that idea and produced that idea. Then there was another set of observations and experiments involving George Beadle and Edward Tatum. And together, they were working on bread mold, or Neurospora crassa, and they were looking at the inborn errors of metabolism associated with bread mold. And so they started looking at the various mutant strains of bread mold. And by looking at these mutant strains, they were able to have a closer look at the metabolic pathway responsible for making of the different amino acids. And specifically, they were looking at the amino acid arginine. And so they were able to find out that one or more of the enzymes that were present in that biochemical pathway towards the synthesis, the making of the amino acid arginine, were defective. So they were able to actually show that. So they took this idea of Garrett's one step further, and they showed that each gene, in fact, does encode for a certain enzyme, and that if you have a gene defective for an enzyme involved in, for example, the synthesis of arginine, then you are going to have that type of mutation leading to a arginine deficient strain. So this was, again, further studied by another scientist, and this time he was studying this phenomenon in humans in the disease sickle cell anemia. And what you might or might not know about sickle cell anemia is in sickle cell anemia, the red blood cells don't have the typical shape that you associate with red blood cells. And I'm just trying to see if I can open up your textbook. I think it has some pictures related to that. And by the way, all of the material from this section is reviewed as material that's covered in chapter 5 of your textbook. So let's see. Okay, yeah, so here is the shape of a sickle cell, and here's a shape of a normal red blood cell. And so, so Vernon Ingram was looking at sickle cell anemia. And what you have to remember is that in red blood cells, there is hemoglobin, and hemoglobin is a protein that actually is made up of 2 types of polypeptide chain. And there are typically 2 types of it. There's 2 of type alpha and 2 of the type beta. And he discovered that when one of the amino acids that make up the beta chain of hemoglobin is substituted for a different one, specifically when you have a valine amino acid in the beta chain substituted to a glutamate, that this leads to a different conformation for that polypeptide chain, one that leads to the red blood cell having that sickle cell shape. So he really then really connected that hypothesis of one gene, one polypeptide, and more significantly, he linked a human hereditary abnormality such as sickle cell anemia to the single alteration in the amino acid sequence of a protein, and single alteration being a valine to a glutamate alteration. And this is significant because there are many hereditary diseases, hemophilia and cystic fibrosis being certainly famous examples of them, that can be traced back to alterations in a single gene. So this entire process of starting with DNA and DNA being transcribed into mRNA and then being translated to a polypeptide, which then eventually folds into an overall protein, this is known as the central dogma of molecular genetics. So this is really central to molecular genetics, that you have DNA, and through transcription, you get mRNA, which then leaves the nuclear envelope, enters the cytoplasm, where it is translated into a polypeptide, which then adopts a specific three-dimensional fold and becomes an actual working protein, carrying out a specific function, whether it's an enzyme catalyzing a certain reaction, or it's a transporter protein, transporting things from one side of the membrane to the other. It's a protein that actually carries out a specific function, be it a structural protein, a catalytic protein, a transporter protein. It has a special function in the cell. And if you have an alteration in this polypeptide sequence in the amino acid, it oftentimes, sometimes actually, I should say, it doesn't affect the protein much, and then other times it really distorts the protein shape in such a way that it no longer has its original intended function. So we've already talked quite a bit about the basic structure of the RNA molecule. We've talked about how RNA, just like DNA, is a carrier of genetic information, but that in a way it's actually different from DNA, in that the RNA molecule has actually got three different forms that perform three distinct functions in protein synthesis. You have the mRNA, and the M, this lowercase m in front of it, stands for messenger RNA. And messenger RNA, mRNA, is responsible for making a copy of that genetic information that is found in DNA. And once it makes that information, and once it makes that copy is then information that is able to move from the nucleus out to the cytoplasm, where then the actual protein synthesis can occur. The other type of RNA molecule that we find in the cell is the tRNA. And the tRNA is supposed to remind us that we mean transfer RNA. And this transfer RNA is responsible for supplying amino acids in the correct order as the polypeptide chain is being made. And finally, we have ribosomal RNA, or rRNA, and that actually plays a structural role. It's a part of that whole ribosomal complex, and it helps in the synthesis of the polypeptide chain as a part of the ribosome complex. Now, to review, many of you might be familiar with this idea, but we're going to review the genetic code a little bit more. As you know, polypeptide chains that make up proteins, these polypeptide chains are made up of subunits of amino acids that are all joined together. So if we go back, for example, to this picture, to this representation, this is a polypeptide chain, and each one of these little blobs here along the chain represent a different amino acid, and those will fold up to give you an overall folded protein. Now, when we're thinking of any given protein, any protein that you might think of, it would be different from other different types of protein in both the number and the order of the amino acids that make up that polypeptide chain. And this is the information that is encoded in DNA. This information is provided in the DNA code. And as a result of this, really, you can provide an infinite combination of amino acids and create an infinite number of proteins, really, with various functions. And evolution has certainly taken care of that. And altogether, as you remember, there are 20 different amino acids that can potentially go into making up a chain. So really a great range of variation is possible. And because we have such large number of amino acids to encode, you have this 3-nucleotide code that is required for each amino acid. So this set of 3 nucleotides that I've shown here in this chart, these are each referred to as a codon. So for example, if this UUU represents the amino acid phenylalanine, CUU represents leucine. And as you can see, for example, in leucine, there's quite a bit of redundancy in the codon, where you have some amino acids that have more than one codon. So any of these, CUU, CUC, CUA, CUG, any of these will give you a leucine, in addition to these two up here, UUA and UUG. So there's also quite a bit of redundancy built into the code. And so this code chart here shows you the codon for all of the different 20 amino acids. And what you can see looking at this chart is that here highlighted in bold is the start codon. And this is the codon that that's recognized by the ribosome to mean the beginning of the amino acid chain. So this is always the start. So all chains begin with AUG, the mRNA, and it has a MET as the first amino acid, a methionine. And as far as the end of the amino acid chains, there's actually 3 possible stops. So this is one, and here's the other 2. There's 3 possible codons to let the ribosome know that this is the end of the chain, and to stop adding amino acids after this point. So what happens for transcription? And transcription is another word for copying. So transcription is that first phase of our protein synthesis. And during this phase is where we get a molecule of mRNA made. And transcription can further be broken down into 4 distinct stages. Stage 1, which is initiation, followed by the elongation, followed by termination, and finally there is a 4th stage called post-transcriptional modification. Now let's go through each one of these steps one by one, and we can take a look at that. And so I really don't want to be jumping back and forth. So let me just see if I can just bring up maybe this picture, and maybe we can just talk about it. But all the notes are are there. So so just to start talking about the first stage, stage 1 is, let me just put it down a little bit more, initiation here. So initiation is when you have this enzyme called RNA polymerase binding to a certain region on the DNA, which is in front of or upstream of the actual gene that is being copied. So it's upstream of it, just ahead of it. And this region that the RNA polymerase binds to is called the promoter region. And the promoter region indicates where that RNA polymerase should bind and start copying or transcribing the information. And this promoter region usually consists of a few hundred base pairs that are typically rich in the nucleotides adenine and thymine. So this is an AT-rich kind of a sequence. So if you're ever looking at a sequence of DNA and you find a stretch that is almost purely, or a good amount of it is made out of A's and T's, that is probably a promoter region and a site for RNA polymerase to bind. And it's typically a few hundred base pairs long. And if you think about it, I'm trying to think if I might have a picture of it further up ahead as to why is it that this region would be rich in adenines and thymines. And it has to do with the way these two nucleotides base pair with each other. Aha, here's a good picture. Okay, so if you look over here, you can see these represent the hydrogen bonds that form across the two strands of the DNA. And if you look between A and T, you have 2 hydrogen bonds. But between C and G, you have how many? 3 hydrogen bonds. So if evolution were to look for something that allows RNA polymerase to bind and make it easier to kind of unzip and open up this region of DNA and not take such huge amount of energy, nucleotides that are joined by 2 hydrogen bonds are not as strongly bound with each other as 3 hydrogen bonds. The 3 hydrogen bond, the bond between C and G, is a stronger set of hydrogen bonds than the A and the T. These are only 2, this is 3. So this are only held together by 2 hydrogen bonds. So that means it makes it slightly easier to open up that double-stranded DNA. So I just wanted to make sure to include that, bring up that picture for you as we're talking about the promoter region. Right. So that being the promoter region, an AT-rich sequence that becomes easier to open up. So once this DNA polymerase binds itself to the promoter region, then the next step is for the DNA strand to be unwound and the double helix is disrupted. And what happens, it exposes this template strand, which is going to be what is going to be copied onto the mRNA. So the next step, the second stage, is elongation. And this is where you have it. So here is where polymerase starts to build that single-stranded message, and we're showing it here in this, just going to zoom in a little bit more. Sort of this light green string here, a strand is supposed to represent the mRNA. So RNA polymerase starts to make mRNA using the transcript, the template strand, as the template. And this mRNA is synthesized in the direction of 5' to 3', and in the mRNA, it is the uracil that replaces the T's. So anytime there's an A, it is complemented by a uracil. So in the mRNA strand, you won't find any T's, you won't find any thymine. Instead, you will find uracil. And as elongation proceeds, this RNA polymerase moves along the DNA, making the mRNA, and the part of the DNA that has already been transcribed, has already been copied, is then is released, and it rewinds back into its double helical form. All those hydrogen bonds take care of, essentially, if you can imagine, kind of zipping up that DNA shut again, and falling into its double helical turn. So if you think about it, since this mRNA is being made in complement to that template strand, then really the code that you would read on the mRNA strand would be the same as not the template strand, but what is complementary to that template strand, or the coding strand. So this strand up here is the coding strand, and it has the same code as the one down here. Because if you think about it, look, these two can base pair with each other, and these two can base pair with each other. So this sequence would be the same as that sequence. We call that the coding strand, except that in mRNA, T is replaced with U. All the thymines are replaced with uracil. So this brings us to termination. So as the DNA is transcribed, as polymerase is reading down, so once polymerase reaches one of those 3 termination codon sequences that we talked about, so any of those 3 stops, once it encounters one of those, sorry, that was the wrong thing to say. No, that is for translation. That is for going from mRNA to protein. But once it reaches the termination sequence at the end of the gene, and there's a sequence to indicate where the end would be, at that point, the polymerase doesn't stops carrying out the transcription, and what happens is that since that mRNA that is being made is complementary to that strand, that mRNA comes off. And once the end is reached, that RNA polymerase will recognize that that termination sequence and just dissociates. The messenger RNA falls off, and the RNA polymerase comes off, and it moves off, and it's free to go on and transcribe another point on the DNA, another message. So at this point, it's the process of transcription is terminated, it's complete, but this mRNA is not quite ready. It's not in a form where it can leave the nucleus and enter the cytoplasm. And essentially for that to happen, we need the 4th stage. We need some post-transcriptional modifications. So as the name suggests, these are modifications that happen post-transcription. So after transcription is done, after transcription is done, we go through a series of modifications that then make the mRNA suitable to leave the nucleus and enter into the cytoplasm. So let's talk about those post-transcriptional modifications. So this primary transcript, the first thing that needs to happen to that mRNA is it needs to receive a 5' cap. And this 5' cap consists of a modified guanosine, so a 7-methyl guanosine is put at the 5' end. We call that a 5' cap. This is a modified guanine nucleotide, and its job is to protect the mRNA from getting digested by the nucleases and the phosphatases as it leaves the nucleus and goes out into the cytoplasm. And this also plays an important role in the next stage of protein synthesis. The second thing that needs to happen to this mRNA is that it needs to get a poly-A tail put on the 3' end of it. So at the 5', it gets a 5' cap, a methyl guanosine. At the 3' end, it gets a poly-A tail. And a poly-A tail is essentially a string, a long string of adenine nucleotides. Here we've only shown like 6 or 7. In reality, it's a couple hundred adenine nucleotides long. It's added to the 3' end by the enzyme poly-A polymerase. So as you guessed it, it puts on, this polymerase puts on lots of A's. So one modification at the 5' end, a second modification at the 3' end, and finally, one more modification can happen, which only really happens in eukaryotes. In prokaryotes, this is it. That's all you need to do. In eukaryotes, there's another further step. And here, the original DNA is made up of exons and introns. And what these exons and introns are coding and non-coding regions. So the exons are called coding, the exons are the coding regions, and the introns are the non-coding regions. And essentially these pieces of introns need to be spliced out, and these exons need to be rejoined to give the final message. Now, originally, these introns were thought to be junk DNA. They were not thought to be having any sort of a function, but we know now that there's actually specific functions there in the intron sequences as well. We won't get into that in the scope of this course, but just be aware that these introns, these non-coding stretches of DNA get removed. And these coding portions need to be strung together. If these intron regions were not to be removed, what will happen is that it will just change what is the normal order of the amino acids in the message. So instead of it reading this way, it has all these extra parts stuck in between. So these non-coding regions then are removed by molecules called spliceosomes, whose job is to excise or remove these introns and join the exons. So finally, once these steps have been done, now this mRNA transcript with its 5' cap, its poly-A tail, and in the case of eukaryotes, these introns excised out, now this is ready to leave the nucleus and enter the cytoplasm. So, just trying to see what we have here. I do have a video that I thought if we have time, we can watch together. But we'll leave that for now. And we'll go on to what happens to the mRNA once it leaves the nucleus and enters the cytoplasm. And that step is called translation. So translation is the second part of protein synthesis. It takes place in the cytoplasm. Here, that mRNA message is read, and the appropriate amino acids are put together and strung in the proper order, forming that polypeptide chain, which folds together and makes that working protein. And there's lots of components involved in translation. One of the important ones is tRNA molecule, and the other one is the ribosome. So the tRNA is a type of RNA that is made up of about 80 base pairs that folds itself into this kind of a clover leaf structure. And it folds itself in this way because there are complementary stretches along the way that will base pair with each other, creating this sort of a clover leaf shape with these hairpin structures along the way. So at the bottom arm of this tRNA, there's three sequences at the very end, or three nucleotides rather, forming this anticodon triplet. So this anticodon triplet, for example, here we have AUA. This is complementary to an mRNA sequence. So if you can imagine, if there was an mRNA sequence down here, this would be complementary to that mRNA sequence. So if you can imagine, this would be complementary to a U, because we don't have any T's on mRNA. U would be complementary to an A, and A would be complementary to a U. So this AUA on the tRNA would be complementary to a UAU on the mRNA. And if you can think back to that codon chart, and we can quickly look back and see, okay, so let me just refresh my mind. So the mRNA sequence that this portion would be recognizing would be a UAU. So if we go back and look at our codon chart, a UAU would be a tyrosine. So when the mRNA sequence has a UAU and it is recognized by a tRNA that recognizes it by the virtue of having an AUA, at this end, the acceptor site of the amino acid, it would be a tyrosine. Yeah, and actually it says it right here in this picture that this is a tyrosine tRNA. So at this end, it would be attached the amino acid tyrosine. So then this RNA with its anticodon will read that message, and it would bring along, because it's specific, so each one of these tRNAs is specific to only one amino acid. This tRNA is specific to the amino acid tyrosine. So it will bring along that amino acid. And the enzyme that is responsible for attaching that amino acid tyrosine, or any amino acid, to the end of this tRNA is an enzyme called aminoacyl tRNA synthetase. And the name kind of hints at what it does. It synthesizes, it makes amino acid and tRNAs together, and that's what it does. The other important component in translation, so there's the tRNA molecule, and obviously there would be the mRNA molecule that brings along the message, would be the ribosome. So the ribosome is actually an organelle. It's made up of 2 units, and let me see if there is a better picture in your textbook. Let's scroll down. I'm looking for a picture of the ribosome with its let me see, where is it? Ah, there we go. Okay. So this here represents, this blob represents the ribosome. And the ribosome has these 2 subunits, which come together to form the intact ribosome. And the 2 subunits are known as the large subunit, which is the larger one, and the small subunit, which is the smaller one. And on ribosome, so yeah, it binds to the mRNA, and here's the ribosome clamped over the mRNA. And as the tRNAs come, this chain, this polypeptide chain is made and elongated. So now let's go back to, yes, and that picture that I was showing you is straight from your textbook. You can look at it. And we can also look at this picture here, but I think I might like the one that I've got a little better. I might show you that one better. So on, on the ribosome, you have 3 different binding sites that are shown here. One site is a site that binds the mRNA transcript. So one of these sites is binding this mRNA transcript, or you can see maybe more colorful in this picture. And the other 2 sites bind tRNA molecules that come in. This represents the other 2 sites, the P site and the A site. And here is like 2 tRNAs that are wedged right in there. So the first one of these sites, the P site, that's the site that holds the growing amino acid chain, or the peptidyl tRNA binding site. And peptidyl, like peptide, polypeptide, you can think of it as that way. The P is where the polypeptide is growing, and you can see it here, it's growing with more amino acids being added on. And the second site is known as the A site, or the amino acid tRNA binding site. Sometimes it's also known, A is for the acceptor site. And the acceptor site holds the tRNA that is bringing the next amino acid that's in line, that needs to be added to that growing chain. So these 2 subunits of ribosome, the small and the large subunits, stay separated in the cytoplasm and only come together when there is an mRNA transcript available to be translated. Then they come together and are form together this unit. So there is a process to to translation. And just like we did for transcription, we're going to divide it up into 3 main steps of initiation, you guessed it, elongation, and finally termination. In step 1 of translation, so translating the mRNA into a polypeptide sequence. So once that modified mRNA, that post-transcriptionally modified mRNA with its 5' cap and its poly-A-tailed and all the introns excised out, once that modified mRNA transcript arrives from the nucleus into the cytoplasm, the initiation step begins. And in this step, the large ribosome subunit that is free, is not bound to the small subunit at this point, the large subunit becomes attracted to to that 5' cap of the mRNA and binds the transcript to its binding site on the ribosome. And once this happened, this prompts now that the small ribosome subunit needs to bind and to kind of sandwich that mRNA transcript between the two subunits. And at this point now, this fully formed ribosomal complex moves now along the mRNA transcript in a 5' to 3' direction. So it moves along in this direction. And until it recognizes that start codon. And the start codon now is that codon that we were noticing on the codon chart. So once it recognizes that start codon, it knows that it should start, it should be starting translation. So here in this case, the tRNA that is carrying methionine is attracted to that complementary codon on the mRNA transcript, and it binds to that P site. This would be the P site. And now the amino acid chain can be extended and to be built on, so we can move on to elongation. So the second step, elongation, excuse me, once that methionine tRNA is present, let's back up a little bit and look at that picture as a whole. Where is it? There we go. So once that methionine is present then in the T site, in the P site, sorry, then the next codon, which comes in at the A site, is read, and the appropriate tRNA comes in and binds to its appropriate site. And the enzyme peptidyltransferase catalyzes the formation of a peptide bond between these two amino acids, for example. And when that bond forms, then that amino acid, so let's say this methionine, imagine it was all the way down here, that one gets released from its tRNA, and then that tRNA dissociates from the site, from the P site. And now both of these amino acids, in this case, I mean, the chain is longer, but imagine you only had the two because you're just starting out, are attached now to this tRNA that is sitting here in I'm sorry, to this tRNA that is sitting now in the A site. And then the ribosome essentially shifts down one codon frame. And the tRNA that's carrying this growing chain moves now into the A site. And again, a peptide bond forms, this time between amino acids with the next amino acid that comes, between amino acids 2 and 3, and it just keeps going around in the cycle as the ribosome moves on, and as the whole peptide chain is transferred to the next tRNA, and that peptide bond is formed. The tRNA dissociates, and the ribosome is ready to read the next amino acid and moves on. The next tRNA carrying the next amino acid will come and bind to its recognition site here, and so on and so forth in this cycle, which uses the energy of GTP. So 2 GTPs are used here, 1 over here. This cycle keeps on continuing until finally the entire amino acid chain is strung together, and you hit a stop codon. And the stop codon indicates that you need to stop. So what happens when you reach the termination, the end, and you need to stop? This is what happens. So let's say there's the stop codon, and this star indicates that last polypeptide, that last amino acid. So, sorry, not the star. This star represents whatever was the last amino acid, and this is the stop codon here. So here in this case, there is no tRNA that codes for the stop codon. So when the ribosome comes to the stop codon, what comes into here is a release factor. It's not a tRNA. It's a release factor that moves into this A site. And when the peptide bond tries to form here, it's not able to do so because there is no amino acid there. And this causes the amino acid chain to simply just come off and float away from the ribosome. And because of the primary structure of the polypeptides, it will immediately start to form and adopt its proper shape. And once this amino acid chain has come off, then that prompts the dissociation of these ribosomal subunits and the mRNA transcript, which is now able to go on and be transcribed again. And the ribosomal subunits are free to bind to the mRNA again, back again at the start codon, or perhaps a different mRNA. Anyhow, once it's done, these things dissociate and are free to bind with another mRNA transcript and translate it all over again. And there's another video that if we have time, we can watch together. So that is how the process of protein synthesis goes from DNA all the way to through transcription and translation. So the formation of that protein is a pretty complex process. It requires quite a lot of cellular energy. And so it wouldn't be very efficient if every single gene was transcribed and translated all the time, right? Because you don't want to be transcribing and translating genes that you don't really need. You don't want to be making proteins that your cell doesn't require and it's not necessary to have around. So there are some genes called housekeeping genes, and those are the ones that are constantly active and need to be producing proteins that need to be continuously present for the cell to function properly, but many of the proteins that are present in the cell aren't necessarily needed all the time. They're only needed in certain times or certain cell types under certain cell circumstances. So in these cases, it would be more efficient if the cell could have a mechanism in place that indicates when protein synthesis can take place at some point during the process. So this type of gene regulation would allow the organism to control specific genes depending on what its requirements are. And there's 5 points of control over gene expression in eukaryotic cells. And those are pre-transcriptional control, transcriptional control, post-transcriptional control, translational control, and finally, post-translational control. We're going to talk about each of these separately. Actually, maybe we should just zoom in on this picture here, and we can talk about so pre-transcriptional control. So it would be here at the level where the cell is able to control just how much DNA gets exposed to the transcription machinery, to the transcriptional enzyme. And because DNA is actually quite highly folded and condensed, in some areas, it's wrapped around tightly around proteins called histones, and forms the structure chromatin when it's wrapped tight around histones. These very condensed regions are generally not very accessible to the transcription enzymes. And so transcription does not happen on these areas, while it can happen in areas that are more loosely folded and are more accessible. So this is at one point where you can have pre-transcriptional control. So by regulating which parts are tightly bound and which parts are loosely exposed, you can exert control even at the pre-transcriptional level. Or you can have gene regulation, and that at the point of transcription, you can have transcriptional control. So in this case, the cell is able to control whether or not that exposed part of that DNA will be transcribed or not. And in the case of eukaryotic genes, there are proteins present called transcriptional factors. And these proteins need to bind bind to that promoter region of the gene even before DNA polymerase can bind and begin the transcription process. And there's other control elements that can affect how well that transcription factor works and can alter the rate at which transcription even occurs. So that's another part. You could also have post-transcriptional. So after you've transcribed and you have your RNA message before it gets modified, you can have post-transcriptional control. And in this case, the cell can control the rate at which the unmodified, so the pre-mRNA, this unmodified mRNA transcript gets modified through post-transcriptional modification into the final finished mRNA through the addition of the 5' cap or the poly A tail. It can control whether these additions occur or it may not occur, in which case, without these modifications, the mRNA is not able to leave the nucleus, or even if it happens to leave the nucleus without those modifications, it gets quickly digested without that protective, without its protective 5' cap and its poly A tail. So in this method, this mRNA might still get made, but it never makes it outside into the cytoplasm, where it will bind to the ribosome and get translated. So there's control even at this level. Finally, there is translational control, meaning the cell is able to control just how quickly and how often that mRNA gets translated into its polypeptide chain. There's regulatory proteins in the cytoplasm that can bind to that 5' cap of the mRNA and prevent, or at least slow down to a certain extent, the ability of that mRNA to bind to the ribosome. So there's translational control, control of just how quickly and how often can that mRNA get translated into this polypeptide chain. And finally, the fifth one is this post-translational control. So if the protein needs any additional processing before it becomes fully functional, this can be either slowed down or eliminated altogether. This can get either slowed down or eliminated together altogether, or you might have, I'm sorry, somebody's dropping off. Who did we lose? Anyhow. And yet there's other types of factors that can interfere with the protein's ability to leave, either if it has to leave the cell or to move to a certain area where that protein is required. There's other factors. And for these types of proteins, it might be that one cell makes them, but they might be exported from the cell, and they might be active in a different type of cell altogether. So finally, the cell can control just how long that protein is functional. It can have different chemical groups added on that may increase or decrease the lifetime of this protein and might mark it for degradation or give it further longevity. So just like eukaryotic cell, prokaryotic cells also use some of these kinds of methods, but because these are single-celled organisms, not all of these methods are utilized to that same extent. And in prokaryotes, the mechanism that's most often used is the transcriptional control. It's at the level of transcription. Now we're going to look at an example of the lac operon. And we're going to go through this and understand this as an example of how a gene goes through this kind of regulatory control. So what the lac operon is, and this here represents the diagrammatic representation of the lac operon, is basically a group of genes located on a bacterial chromosome that contains a series of structural genes, a promoter region, and in between the promoter region and these genes that encode some structural proteins is this sequence called the operator. And this operator acts as a regulatory sequence. So these structural genes within this operon here, these code for different proteins that are involved in lactose metabolism in this type of bacteria. And lactose, if you remember, is a type of sugar. It's a disaccharide that is found in milk. And it's a disaccharide, meaning that it is made out of one glucose molecule and one galactose molecule that are joined together. And since this sugar isn't always available to bacteria, for example, to E. coli, the control of these, the expression of these genes needs to be controlled, needs to be regulated. It wouldn't make sense to produce these genes at the time where there is no lactose available as a source of fuel. So this needs to be regulated. And these genes are called LAC-Z, LAC-Y, and LAC-A. Essentially, LAC-Z, what you need to know is codes for an enzyme beta-galactosidase, which is able to break down lactose. LAC-Y codes for beta-galactosidase permease. So these are all beta-galactosidase gene. This one is for an enzyme that is able to break down lactose. This is encodes for an enzyme that makes the membrane more permeable to lactose, so the lactose can enter the cell more easily. And LAC-A, don't need to worry too much about it. And so these three structural genes. So if you don't have any lactose present in your media, suppose you're growing bacteria on a petri dish, or there's some bacteria and lying around and doesn't have any access to lactose, you don't need to express these genes. In this case, you have a repressor protein called the LAC-L protein. Right here. And this LAC-L protein, excuse me, its job is to bind to this LAC operator. And because there's a certain amount of overlap between the promoter region and the operator region, when this LAC-L binds, it actually, excuse me, the binding of this repressor protein covers part of this promoter. So if you can see in this picture, they're trying to show it too. So once this repressor protein binds to the operator sequence, it's kind of bound to part of the promoter region too, part of the promoter sequence. And by doing so, now RNA polymerase is not able to fully bind to the promoter region. It's kind of blocking it. It prevents RNA polymerase from binding to this promoter. And since RNA can't bind to the promoter, it can't go ahead and transcribe this gene. But if, on the other hand, say you had lactose, see this green ball is supposed to represent a molecule of lactose. If you had lactose present, then you need to be able to move this repressor protein off of the operator because you have lactose present. You want to be able to express all of these genes because these express enzymes and proteins that allow you to use that lactose as a source of food. So you want to get this repressor off and get things going. You want mRNA to be able to bind and kickstart transcription. So what happens is lactose itself comes and binds to the repressor protein and acts as an inducer by binding to this repressor protein. And when it binds, it actually causes a conformational change, a change in the shape of the protein, so that it can no longer bind to that operator. See, now that it's bent in this shape, for example, it's got a different shape, it can no longer fit into this operator sequence here. It can no longer bind to the operator. So once it binds to this and removes it and gets it off of the operator, then now the genes can become translated and made into all of these working enzymes, the beta-galactosidase, the permease, and the transacetylase. Anyhow, all of these are enzymes and proteins that you need in order to metabolize lactose. And since you have lactose, lactose binds to the to the repressor, gets it off. RNA polymerase is able to bind and carry out transcription. And so now you have your mRNA message transcribed. So that is the story of how the lac operon, for example, is a good story of gene regulation through the presence of lactose itself, acting as a regulator of the expression of this gene, these set of genes. So we've talked about and we touched on the idea that you could have a change in a certain amino acid that can cause enough of a change in the protein sequence. So that change in the amino acid ultimately comes as a result of a change in the genetic material. And those are known as mutations. So when there is a change, sometimes a permanent change in the genetic material, that permanent change is known as a mutation. And mutations can oftentimes be very harmful, have very serious effects. Sometimes they can have no effects, and sometimes they end up having some sort of a positive effect, depending on exactly what type of mutation has taken place. In many cases, these mutations are caused by various agents in the environment, such as radiation from X-rays or gamma rays. Those cause quite a lot of damage to DNA. Sometimes there are physical mutagens that can actually break through the DNA strand and cause random changes in the nucleotide sequence. UV radiation from the sun is not as strong as, say, X-rays or gamma rays, but it can still, excuse me, cause damage. And that's what happens in the case of skin cancer. It's UV damage from the sun, damaging the DNA and skin cells and having very deleterious effects, very serious effects. There's other types of mutagens that are chemical mutagens. These chemicals are oftentimes able to directly interact with the DNA molecules. Sometimes these mutagens, these chemical mutagens, have structures that are very similar to the nucleotides themselves and actually become incorporated into the DNA during replication. And if that happens, DNA polymerase can no longer recognize, obviously, the sequence, because it is not any of the nucleotides that it is learned to recognize, and it causes confusion. And when it's trying to determine what nucleotide is it supposed to be on and what nucleotide it should insert, it causes maybe some missense or nonsense mutations. And yet there's other chemicals which can intercalate which means it can insert themselves in between the nucleotides, in between the bases of the double helix, and interact with those nucleotides in a way that during replication, polymerases may miss a nucleotide or put in extra nucleotides as it's trying to read this distorted code. So there's plenty of things that can happen to bring along mutations into the DNA. But there's two main categories of mutations that affects individual genes, and those are point mutations or frame shift mutations. So in point mutations involve a nucleotide substitution, where you have one nucleotide replaced by another. And in this type of mutation, only one codon is affected. So let me see if I can find a good example of this in your textbook. Let's see, do they talk about mutations? Types of mutations? Yes, okay. So there's a little picture here. They show a point mutation. Okay, so for example, a point mutation would be instead of a G, it gets a C, for example, where only a single codon is affected. Or instead of AAC, it becomes GAC, so a single codon is affected. So that is a point mutation, where there's only one codon affected. That type of mutation can be a silent mutation, meaning that nucleotide substitution changes the codon, but that codon still codes for that same amino acid. If you remember, we said earlier on that there's some redundancy in the codons, and some amino acids have more than one codon. So this means that still, even though the nucleotide has changed, it's still encoding for the same amino acids, or the order of amino acids is essentially unchanged, and the protein is not affected. And really, this is beyond the scope of this course, but just to let you know that there's been some evidence indicating that there can still be an effect, even though it might only be a silent mutation. It may not be so silent. Because sometimes when there's more than one codon encoding for an amino acid, some codons are more commonly used than others. So if the silent mutation substitutes for a more commonly used codon for a less commonly used one, then the cell can have some difficulty finding that type of codon, and that substitution will eventually have some sort of an effect on the folding of the protein, which can affect the protein function. But this is an area that's still being quite a bit of research is being done on it, and still trying to understand why this this happens. So that's a point mutation that can be actually a silent mutation, can have no effect on the actual amino acid. The other type of point mutation could result in a missense mutation. And that is when that nucleotide substitution causes a change in that codon that actually makes it code for a different amino acid altogether in the protein sequence. So let me see if there's an example of a missense mutation here in, for example. So for example, here is your normal RNA strand. The mRNA reads AUG, AAC, CCC, ACA, UAA, which means stop. So suppose there was a missense mutation, a missense mutation, which might change this threonine, which is encoded by the codon ACA. Suppose there's a point mutation that changes it from ACA to CCA, and instead of coding for a threonine, it now codes for a proline. Well, this is a missense mutation, because even one single nucleotide substitution has led to a different amino acid being inserted here. So even one change could have a serious effect on the proline. For example, the case of sickle cell anemia that we discussed, where you can have a change from a normally shaped red blood cell to a sickle cell shaped blood cell caused by a change in a single amino acid in that whole hemoglobin protein. The other type of point mutation that you could have is one where you could have a nonsense mutation, where you have a change in the nucleotide that changes the codon in a way where it changes an amino acid into a stop codon. For example, in this case, this is a nonsense mutation. So say, for example, where you normally have AAC, now you suddenly have a UAA, and that codes for a stop. So this means that the protein stops being made prematurely. And so now it's kind of cut short, and it stops being made only up until this point. And depending on where the change occurs, that protein could be missing a small portion or the majority of the amino acids that form it. In case of this protein, look, it's being stopped before it's even made. But suppose this was a very long protein, and maybe after almost all of it was made, there was a nonsense mutation that caused it to stop. In that case, we call it, it's a truncation. It's being cut short. And maybe it might be still enough there that that protein might function, maybe not function to its highest efficiency, but it still may have enough function, or it may not. It may be that it is missing crucial parts of that polypeptide chain that allow for the correct fold. It really is specific, but in nonsense mutations, the point mutation creates a stop codon, causing the polypeptide to end before it's supposed to. Sometimes you can have a frame shift mutation. So that means maybe there's an insertion or a deletion. So for example, let's look at this sequence here, where you have methionine, asparagine, proline, and then the proline, after the proline comes ACA. But suppose this third C of the proline was knocked out, was somehow skipped during transcription, then that causes a frame shift. Then that causes this next A to move into this place if this C were gone. So now we have CCA, which oddly enough still codes for a protein, for proline, but then it's followed by a CAU, whereas it used to be an ACA coding for a threonine. Now it codes for a histidine, and then everything after that, it's a frame shift. So everything after that, the entire reading frame is shifted, and that all the codons beyond that point where that nucleotide was affected, everything beyond that, the whole sequence is affected. So this is a frame shift mutation. Or maybe instead of a deletion, you get an insertion. Maybe you get, as this was being transcribed, a U gets mistakenly inserted here in here. And again, this causes a frame shift mutation, which may affect all the amino acids beyond, or it may insert a sudden stop. So there might be a frame shift causing a nonsense mutation. And sometimes the mutation of the DNA can actually involve larger groups, larger stretches of the nucleotide. In this case, we call it a translocation, or an insertion, or a deletion, or an inversion, where you can have big chunks. Let me see if they have pictures of it here. No, they don't. But if you can imagine, entire segments might be taken out or put in backwards, as is the case in translocation, as is the case in inversion, or entire chunks may get removed and inserted from one area into another area, as is the case in translocation. So, so any of these types of changes can have extreme effects on the organisms. It's thought that mutations are also at least partially responsible for the various evolutionary changes that have taken place, and may have been the driving force behind the formation of new species, where through mutation, new traits come up, and if they give that that organism a certain advantage, especially at a time where all the other organisms might be at a disadvantage, then this mutation may actually take hold and give the offsprings of this organism even a better chance at survival. And that's at least partially how evolution may have taken place. All right, that brings us to the end of our lesson 10. We have a few minutes if we can go back and we can watch some of those YouTube videos together. So there was one here that we can watch, and there was another one further up. I think it was just these two, as far as I remember. Yeah, we can go ahead and add these two. We can watch these. I'm sorry, I didn't have these on cue. So I'm just going to quickly bring them up.
Have you ever been at your nursing lab at your school, watching your faculty demoing?
Okay, I'm just going to wait until this, there we go.
Now that we've covered DNA replication, let's talk about transcription. The first thing you need to know is that transcription has nothing to do with cell replication processes such as DNA replication, mitosis, or cell division. So, what is transcription and why is it necessary? Well, transcription is the first step in the process of using the genetic code in DNA to synthesize or build all the different proteins in your body. One problem with synthesizing these proteins is that the instructions for making them are in the DNA, which is located inside the nucleus. But the place proteins are always made is outside the nucleus, either in the ribosomes floating in the cytoplasm or in the ribosomes embedded in the rough endoplasmic reticulum. So, how does the genetic code for synthesizing proteins get from the DNA to the ribosome? DNA uses a messenger called messenger RNA or mRNA to carry the genetic code from the nucleus to the ribosome. The process of building this messenger RNA is called transcription. Now, let's see how transcription happens. Transcription begins when an enzyme called RNA polymerase attaches to a segment of DNA called a gene. A gene contains the code to build a specific protein, which is a macromolecule made up of a sequence of amino acids in a specific order. And within a gene, the specific order of nitrogenous bases dictates the order of amino acids that will make up the protein. Each group of 3 consecutive bases in the gene is actually a code for a particular amino acid. As a result, each group is referred to as a codon. RNA polymerase causes a particular area of the DNA helix to unwind and separate into 2 strands. One of the strands, often called the template strand, is the side of DNA that is read or transcribed by the messenger RNA. The other strand of DNA, often called the non-template strand, isn't transcribed by the messenger RNA. So, how are DNA instructions transcribed into messenger RNA? Well, using the template strand as a guide, RNA polymerase uses the base pair rule to assemble free nucleotides in the nucleus into a complementary strand of RNA. For example, RNA polymerase reads the DNA base thymine on the template strand, then binds it to a free nucleotide containing adenine. This process continues with cytosine binding to guanine and guanine binding with cytosine. Remember though, RNA will never contain thymine. So, whenever RNA polymerase sees adenine on the DNA template strand, it pairs adenine with uracil. By using the template strand of DNA as a guide, the genetic code from the non-template strand of DNA has actually been transcribed into messenger RNA. When transcription is complete, the messenger...
I just want to bring your attention to that last bit again. So what I want you to notice that this is the template strand, and this mRNA has been made complementary to this template strand. But if you notice, this non-template strand corresponds to the sequence on the RNA and the mRNA, with the exception of every time you see a T, you have to think of it as a U, because on mRNA, there's no T's, it's U. So this non-template strand is also, the other name for it is the coding strand, because you can read the code on the mRNA from that strand.
Has actually been transcribed into messenger RNA. When transcription is complete, the messenger RNA, which is small enough to fit through a nuclear pore, takes the genetic code out of the nucleus to the ribosome, the site of protein synthesis.
Of course, you guys remember that it does so once it's been transcriptionally modified, meaning that it should have its 5' cap and it should have its poly A tail at its 3'. And in the case of eukaryotes, all non-coding chunks have to be excised out.
The process of actually building the protein at the ribosome is called translation, which we'll cover in a separate video. To summarize, transcription is the process of transcribing or copying the genetic code for building a protein into messenger RNA. A gene is a segment of DNA containing the instructions or code for building a protein. A codon is a group of 3 consecutive nitrogenous bases in a gene containing the code for a specific amino acid in a protein. RNA polymerase unwinds the strands of DNA in a gene. The template DNA strand contains the complementary bases that need to be read to generate messenger RNA. The base pair rule is followed when assembling messenger RNA. Messenger RNA is actually a copy of the DNA non-template strand with uracil substituted
In
for
order
thymine.
for our bodies to function, we need to
Okay. So now I've got the next one. And I'm sorry, we're gonna have to
supply them with a variety of nutrients we
watch
get from
this.
our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into 2 processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. Proteins are needed for most physiological functions of the body to occur properly, such as breaking down food particles in digestion, and the processes of transcription and translation make the production of proteins possible.
All right. That brings us to the end. I know we went a few minutes over time. Thank you, all of you, for still being here and listening to your complete lesson. So on our next lesson, we're going to continue on with lesson 11, where we're going to talk about biotechnology, gene modification, and some of the tools that are available to us as scientists that we can use to manipulate DNA. All right, if there are no questions, I'll see you guys next time, okay?
Thank you. Good night.
Thank you, bye-bye.
Miss, I just have one question.
Yeah, 1 second.
I just want to know when our project is due.
Yeah, I haven't, it'll be due closer to the end of the course. I haven't actually arrived at a time yet, but I should have that for you guys soon.
Okay, thank
Okay.
you.
Right, no problem.
Okay, bye, Miss. Have a good night.
Thanks, you too, Natalie.
Okay, bye.